Hi, I'm Jessica, and I just graduated with a degree in environmental engineering.
My name's Garfield. I just graduated from UC San Diego with a degree in biology.
And I'm Ava. I'm going to be a third-year environmental engineer at UCI.
And today we're going to talk to you a little bit about the ecology, specifically of the wetlands and biofilters that we visited when we were over in Victoria, Australia. So as Stan pointed out, Australia recently went through a huge drought known as the Millennium Drought that started in the late 1990s and lasted more than a decade. Particularly Melbourne, which is where we visited, right on the southeast tip of Australia, was hit one of the places that was hit the hardest by the drought. And the drought severely depleted their drinking water sources, so they were forced to turn to alternative water sources for uses that were uses other than that of drinking water. And one of the things that they turned to was recycled water. Recycled wastewater would be one. That's like this down here. The Western treatment plant treats wastewater, and then also wetland and biofilters to treat stormwater for uses such as toilet flushing and things like that. And then also, we saw this implemented more on the outskirts of town, or of the city, rainwater tanks, which are right here. So a lot of residents will collect rainwater and connect the rainwater straight into their homes and use that for toilet flushing, for doing their laundry. And one woman we talked to actually said that they use their rainwater for everything from bathing to anything that's not drinking water. And but when you have these wetland and biofilter systems and things like that, there is a potential human and ecosystem health risk. It's hard to tell whether these systems are working optimally just by looking at them. So that's why we went over and did the testing that we did. You can see here, this is Garfield. He was in the water trying to collect sediment core samples, and this is actually the inlet of one of the sites that we visited, and you can see that there's a lot of trash in the water. The water was very turbid, and when we were there, we smelled sulfur in the water, which is an indication that there was very low dissolved oxygen levels, which means the water quality was relatively poor. So one of the main things that we were focusing on is the ecology. When we visited these sites, we saw a lot of water birds and wetland plants, which means these wetland sites are not only providing water treatment, but they're also providing habitats for local species. And we looked also in the benthic layer, which is a couple centimeters of sediment in the water. And we looked at the macroinvertebrates there. And the two main types of macroinvertebrates that we found were the water boatman and the chironomid. And our project focuses mainly on the chironomid. And what we wanted to look at is the chironomids as indicators of water quality. Now, a chironomid is a larval stage of a midge, which is a non-biting fly. They thrive in anoxic conditions, which means low dissolved oxygen in the water due to the hemoglobin in their bodies, which is what turns them red, and it allows them to efficiently use oxygen in the water, which allows them to live in places that have virtually no oxygen. They also feed on organic matter and are associated with degraded wetland conditions, which is why we chose to focus on the relationship between chironomid abundance and overall water quality for our project. So we looked at specific water quality parameters, and Ava is going to shed a little bit more light on what we did.
So we used several different techniques to collect data for water quality. We visited 2 biofilters and 3 constructed wetlands in Melbourne. The way we found out how many chironomids there were was that we used a pool net, as you can see Dr. Engineering is using, and we would collect around 1 meter squared of water and sift through it, and we would see what kind of macroinvertebrates were in there. We would put the number of macroinvertebrates into certain bins, so the bins were 1 to 5, 5 to 10, 10 to 20, and we would collect the number of chironomids by that method. We would find out chlorophyll concentrations by water sampling and vacuum filtration and spectrophotometry. We measured dissolved oxygen and temperature with an Orion sensor, and we measured conductivity, turbidity, oxidation reduction potential, and pH with a Horiga sensor. The are showing the three of us doing 3 different techniques of data collection. On the left, I'm comparing a water sample for total suspended solid filtration. In the middle, Garfield is using an Orion sensor in the water, and on the right, Jessica is using vacuum filtration with a chlorophyll sample. So after we collected our data, we analyzed it with a software called Virtual Beach 2.3, and this software did a multiple linear regression on the data. We tried to find linear relationships between chironomid abundance and water parameters such as chlorophyll content, dissolved oxygen, pH, temperature, conductivity, turbidity, and oxidation reduction potential. Virtual Beach was not able to find direct linear correlations, and so we did transformations of all these variables and were able to find results that way. So this is the best result that Virtual Beach found. The best correlation with the chironomid abundance was with the inverse of turbidity and the inverse of temperature. There was a negative correlation with the inverse of temperature and a positive correlation with the inverse of turbidity. And in this graph, you can see the x-axis is the sample ID, so each plot had an ID number, which it was referred to, and the y-axis was the number of chironomid. The blue line is the observed number of chironomid, and the black line is the predicted number. And also, as you can see in this graph, there's a huge range of chironomid. Plot 7 has around 150, and plots 4, 5, 11, 12 have 0. And so this huge range was very interesting to us, and there should be a reason why this range exists. And if you can imagine, around 1 meter squared of water with 150 chironomid, that's a huge amount. One thing we found especially interesting is that plot 7 was the Limburg wetland, and that was actually the oldest wetland we visited. So we think there may be some sort of correlation with the age of the wetland and the number of chironomid. The second best correlation that we had with chironomid abundance was with the second order polynomial and the inverse of turbidity. And again, there was a positive correlation with the inverse of turbidity. And now Garfield will discuss what these results mean.
So based on the results that I will discuss, we can theoretically predict the chironomid abundance using either temperature and turbidity or using pH and turbidity. Furthermore, I want to expand on turbidity because it has been shown in 2 of the models, explaining that it's probably one of the more credible of the 3 variables. And lastly, we also did some preliminary classification tree analyses on our data and found that it was the same 3 variables, which is pH, turbidity, and temperature, that came out as the top 3 variables to predict this chironomid abundance. However, we do admit that our data set is very small. There are many holes, few and far between, and that our analytical techniques are not necessarily the best for categorical data. The multiple linear regression and the techniques we used in Virtual Beach 2.3 was primarily geared towards numerical data. But because our BINS data are considered categorical data, we will have to transform it into numerical data, and that can account for some of the errors and the skew, like the differences that we saw in our results. So in conclusion, our study has identified that temperatures, pH, and turbidity are possible indicators of chironomid abundance, and that our data and our methods are insufficient to conclude that these water quality parameters are able to predict chironomid abundance. And in the future, we want to focus on a couple of things. First of all, we want to increase our sampling size and our sampling intensity. We want to get a clear picture of this, and to get a clear picture of this, we need to have more samples and more work done in these areas. Secondly, we want to survey these alternative variables, survey alternative variables, such as wetland birds and the age of the biofilter. As Ava said earlier, Limburg was one of the oldest biofilter there, but there are many that are not that old, and that can provide 2 reasons why chironomid abundance would change. First of all, the older the biofilter or wetland, the more time frame it gives for chironomid to settle and pervade through the system. Secondly, the amount of time that the biofilter and wetlands are exposed to the water can also change the soil composition in the water, allowing to either expand or decrease the range or the habitat of the chironomid. And second of all, we want to explore wetland bird density. And this is because wetland birds could predict on chironomid. And because there are different density at different biofilters or wetland, there may be an interaction going on here that we have not accounted for in our data. Another method we want to change is to use a different statistical tool. We want to use a statistical tool that allows us to evaluate categorical data, and those include generalized linear models or classification tree analysis. And lastly, we also want to look at the biology or the functional role of the chironomid by itself. We want to know what chironomid is doing in the system and how it's affecting the water quality. So with that, I'd like to acknowledge the people who helped with our project, Dr. Stan Grant, Dr. Megan Rippey, Dr. Andrew Mary, and Dr. Sunny Jane. And the graduate students, Alice and Laura, Nicole and Layla for taking care of us, the faculty of University of California, Irvine, and the faculty of University of Melbourne for contributing and facilitating our research. And also, we want to thank NSF for funding our research and making all this possible. So with that, I'd like to take any questions.
Did you look at salinity? Because salinity, I think, also has an impact on chironomid fish, inhabiting its site. And also in terms of wetland birds, take a look also at swifts and swallows. But
Did we look at salinity? Is that the question?
We have the data, but there are a lot of holes in our data because not every plot that we went to we could collect water, or not every plot that we went to we could deploy our sensors. So if somewhere we had a number of chironomid, we didn't have like nitrogen phosphate or something like that.
So salinity wasn't, we didn't have a full profile of it, so we ended up having to take that out of the model when we ran the software.
my guess would be, things, true freshwater is 0.5 or less parts per thousand, and anything probably below about 5 or so would probably have more than things up by 9. I don't know what kind of system you were really dealing with.
These are all freshwater levels.
They were.
Any other questions? Let's thank our